Hi everyone. So my name is Anika. I am a streamer for AP Biology. I am a senior, and today I'm just going to be talking about pretty much how to approach the FRQ for AP Biology and I'm going to be going over the exam changed a little from last year. So we're going to go over a few of the changes to the FRQ, and then we're going to do some practice FRQs, and in that I'm going to give you some tips and tricks that helped me personally when I took the exam last year, and to kind of take some of the stress and some of the scariness out of the FRQ, because the AP Bio exam FRQs sound really terrifying. I used to be scared of them, but then it's not so bad. So just to briefly go over the new format of the exam. Actually, how do you guys feel about FRQs? If you want to drop it in the chat and just, how is it going? Have you done any yet? Have you done any in class? Do you like them? Do you not like them? Are you scared? Are you not scared? You can drop an emoji to show tell me how you feel about the FRQs. And then I'll go ahead and start going over the exam format, and we'll go ahead and get started. So while you guys do that, it's okay, don't be shy. So pretty much the new exam format, just like last year, you still have 1 hour and 30 minutes to complete the FRQ portion of the exam. So you have 1 hour and 30 minutes, and last year we had 2 long free response questions and 6 short free response questions, so our time was a little crunched. But this year, lucky for you guys, you have 2 long questions and 4 short questions. So you have a little bit more time to spend on each. I see what you guys are saying in the chat. That's not very good. So hopefully this stream helps you. So when you get the next one, you can feel more confident about it. So in terms of the long free response questions, they're going to be worth 8 to 10 points each, like a range depending on like what the question is asking. And at least one of those questions is going to be involving like graphing. And I'm going to try, if we have time at the end, to go over one specific like part that involves graphing, because it sounds really scary. And when you read the question, when you're stressed on the exam, you can mess it up in your brain and make some silly mistakes. But in reality, the graphing questions are usually really easy. Like when you just look at them, they're like elementary level graphing. And the short free response questions are each worth 4 points. So you'll have about 10, if you spend 10 minutes on each short free response question, you'll have 20 minutes to spend on each long free response question. So time, hopefully, should not be too big of an issue. So some quick tips before we get started, and then I'll also give you some as we go through. Look at all of the questions first. So go through the exam. When you get your FRQ and you open it, okay, the seal is off. Now it's time for me to start. You don't have to start with number 1. When I did the exam last year, I started like number 4. And I just went based on, okay, I really like this topic. I really like genetics. That's like my favorite topic, genetics, and also I liked viruses. So I went through and I said, okay, I really like this topic and I'm really good at it. So I'm going to answer this topic to kind of get my confidence up, and then I'll go through and answer the topics that I'm not so good at. So it's kind of like a confidence boost, because if you start on a question that you're not confident in and you don't think you're going to do well on, it's kind of a downer, and it'll kind of get you off your game for the rest of the questions. So it's best to start with one that you really know really well, or a topic that you like and you're interested in, you have a lot to say about it, so that way you can kind of get on a roll. Another tip is to just, if it's about a topic, like let's say genetics and it's about, or cells, since we're going over cells now and with our streams. If it's about, for example, cell division, and it's about mitosis, you want to say all the steps of mitosis, you want to get all your keywords in there, because that's what the readers are looking for. When you look through the scoring guidelines, like we'll do in this stream, you'll see that it's like, okay, some keywords, they mention prophase, metaphase, they mention all the phases, they mention this. So you want to try to kind of get all the things that you know on that topic in. But it's kind of a perfect in between, because at the same time, if you say too much and you start almost contradicting yourself, you can get points off. So you have to be careful. But in general, what I would do is I would just kind of write what I think is related to the question. And it's better to write more than to write less. Again, just look at the core topic and what you know about it. And also, so the questions generally will ask you a few things. It'll say identify, explain, predict, compare, contrast, discuss, describe. There are a lot of different words that they use. One big one that they use is justify. So it'll be like, these are we have one in here. It'll be like, these are the results of the experiment. Justify, or a student makes a claim, justify the claim, or you make a claim, justify the claim. Those are really important, because that's especially where they look for keywords. a student makes a claim that this is a plant cell. You need to say it has a chloroplast. It has a vacuole. So you have to say very specific things that they want you to say in terms of justification. It has to be like facts that you've learned in class. That like questions part with justify is seen like all the time. But other than that, identify, explain, predict, those are seen in a lot of your other classes. They're just, as they say, like identify just means answer the question. But explain, discuss, all of that you need more justification for. Okay, so I'm going to go ahead and get started. Let me share my screen. And I will also put the link. I have to remember how to do that, but I will be putting the link in, hopefully. Let me share my screen so you can see. Make sure it's the right one. Okay, so here's my, this is 2018 questions. So I picked a few, and since we're going over cells now in the streams, I decided I'd start with cells, or try to have them somewhat related to cells. But again, a lot of the questions cover multiple topics. So we are going to get into some things that we didn't discuss yet in streams, but I'll explain it as we go. And it's more just about learning about how to approach these questions, you know? Okay, so here you see, this is what the actual exam looks like. So it tells you some instructions up here. And if you don't remember exactly like what you're supposed to do, or if you get scared on exam day or anything like that, these instructions are kind of helpful. Usually when I do exams, I usually have done like a lot of practice exams, so I don't need to read them. But if you need to read them, it's not a problem. Just make sure, so it helps you to know what you need to do exactly. So this is from last year's exam, which had, again, 8 questions instead of the 6 that you're going to have. So the times that they give you here are not entirely accurate for you. But okay, so this is, where is it? We're going to do number 2 because it's somewhat related to cells. So this is an example of a long question. So the long questions, you guys, if you spend, on your new exam, if you spend 10 minutes on the short ones and about 20 on the long ones, you'll be perfectly fine. So the long ones, as you can see, they're pretty long. And they have a lot of different parts. So A, B, C, D, sometimes an E, sometimes it goes even further than that. Whereas the short ones will just be like 1 part or maybe 2. So the timing, when you say 10 minutes on the short ones, it sounds a little bit scary, like, oh, only 10 minutes, but they're not a problem. The ones you really need to focus on are the long ones. Okay, so we're going to read this together, and then I'll give you guys a few minutes to think about it, and if you have any ideas or any questions, be sure to put it in the chat, and I will be very happy to answer it. And after maybe 5 minutes or so of you guys just, brainstorming in the chat, I'll help you guys out. You can go through my thought process, and then we can go over the scoring guidelines. Okay, so here you have this picture. You have to always make sure to look at the picture and analyze the picture. And so here you see the first step in whatever's going on here, and then it splits into 2 steps, and then something else happens. So without reading, you don't really know exactly what's happening, but clearly it's in a cell because you see the cell membrane. Okay. Some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways, figure 1. So that was just an introduction telling you, okay, this is a host organism, there's some sort of bacteria in the organism, and this is a way that the host cell responds. So this is just giving you some context so you know what exactly that picture is that you're looking at. Oh, I have to put the link. I'm sorry. Let me see if I can remember how to put the link in. Okay. I cannot remember how to put the link in, I'm sorry, but this is the 2018 exam, and this is number 2. If you want to find it, it's on College Board. It's super easy to find. I'm sorry. So if someone finds it and they put it in the chat or something, that'd be great. Thank you. Okay, so cells normally produce a steady supply of inactive caspase-1 protein. In response to intercellular pathogens, the inactive caspase-1 is cleaved and forms an active caspase-1, step 1. So that little 1 on the diagram, that's what it's describing. Active caspase-1 can cleave 2 other proteins. When caspase-1 cleaves an inactive interleukin, step 2, so now you look at step 2 and you're kind of understanding what's going on now, the active portion of the interleukin is released from the cell. An interleukin is a signaling molecule that can activate the immune response. So they actually tell you what an interleukin is. Thank you. I just cut my hair. Thank you. So they actually tell you what an interleukin is here. So that's kind of a good thing because now you get a little bit of context. Okay, interleukins start like the immune response. So you have some context for what's happening here. They tell you what an interleukin is, which is really great because you don't have to kind of try to figure it out. Okay, so when caspase 1 cleaves Gasdermin, step 3, so now you're looking at step 3, and you can, okay, I see the Gasdermin is being split or cleaved. The N-terminal portions of several Gasdermin proteins associate in the cell membrane to form large non-specific pores. Thank you for putting that in the chat. If you guys want to open it yourself, this is number 2 on the link that was just put in the chat. Researchers created the model in Figure 1 using data from cell fractionation studies. In the experiments, various parts of a cell were separated into fractions by mechanical and chemical methods. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table below shows the presence of known proteins in specific cellular fractions. Okay, so here you have a table, and it's pretty much telling you where the proteins are located. So what I would do if I saw this table, because it can be a little bit confusing to like digest if you don't really know what you're being asked, I would kind of know, okay, generally the table is telling you where proteins are located. But I wouldn't go too far in trying to figure out, okay, I think fraction 1 is a this, fraction 2 is a this, because I don't know what the question's going to ask me. So I would go and read the questions, or read the question, and as I go, if there's something that I see needs the chart, then I go back up and analyze it in response to the question, because otherwise you're really wasting time. Like, let's say I analyze something that the question doesn't ask me. Time is really important here, so I think it's best to, when you see something like this, analyze it to the best of your ability, know what it's about, but you don't have to go and like know everything about it, because you do have it for reference. It's not like going to be taken away from you. Okay, so read all the parts first, so you can kind of get an idea of where they're going, and then answer. So A asks you to describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell. B asks you to make a claim about how cleaving inactive caspase 1 results in activation of caspase 1. And then it tells you a student claims that pre-infection production of inactive precursors shortens the response time of a cell to bacterial infection. And you have to provide a reason to support the student's claim. C, as you can see, a student claims that the NFKB protein is located in the cytoplasm until the protein is needed for transcription. Justify the claim with evidence and identify 2 fractions where the terminal Gasdermin would be found in cells infected with the bacteria. Part D, describe the most likely effect of Gasdermin pore formation on water balance in a hypotonic environment. Explain how, and then E, explain how Gasdermin pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. So the only one here that I've noticed that is asking about this table would be part C, because it says identify 2 fractions. But other than that, it doesn't look like the table is going to be very important. So going back up, starting with part A, describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin. So if you guys want to put in the chat what you're thinking about, your thought process. So it's saying inhibiting step 3. What would its effect be on the formation of pores and on the activation of interleukin, on the release of interleukin? So step 3 is this one up here. I think you can see my mouse. So step 3, yeah. So step 3 shows, sorry, let me fix that. So step 3 is this step. So what would its effect be if it didn't happen? What would happen to the pores and what would happen to the interleukin? So if you guys want to put any ideas you have in the chat, or any questions, if you think I missed something or if you want something clarified, I'll give you guys a minute. This portion of the question is one that shouldn't take very long. The other portions take a lot longer. So I'll give you guys about a minute to talk in the chat, see what other people are thinking, see if you're thinking anything similar. If you disagree, say you disagree so we can get some discourse going and kind of identify if you guys are having some problems or not. So I'll give you guys another until 7:15, so less than a minute. Don't be scared to talk in the chat, everyone. Okay, so as Shreya said, the pores wouldn't form, but the interleukin wouldn't be affected. So here you have the Gasdermin. If step 3 didn't happen, you are correct. The pores would not form because step 3 causes the pores to form. So without step 3, without all of this right here, this can't happen. It's part of a pathway. And the interleukin would not be affected, so you're right again, because here you see the pathway it splits off. So what happens to one doesn't necessarily happen to the other. So that section was actually pretty straightforward. But you have to describe the effect. So I would say in just 2 sentences, it doesn't have to be too in-depth. So you'd say if step 3 didn't happen or was inhibited, the pores would not form because the Gasdermin isn't created, and the interleukin would not be impacted because it's a separate step, or something along those lines. Okay, part B. Make a claim, as I was saying, they ask you to do that a lot, about how cleaving inactive caspase 1 results in activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to bacterial infection. Provide one reason to support the student's claim. Okay, so for this, you don't even really need the picture. It's more asking about just like general biology topics. But the little paragraph that they give you, that helps more so than the picture, in my opinion. But the picture, I'm sure, can help for some people. So this one, I'll give you guys a little bit of time to think about it. Share your thoughts in the chat. If you have some questions, also put it in the chat. If you haven't gotten like here yet in bio, that's perfectly okay. We're just going over more so the thought process behind how to answer the questions. So if you haven't gotten here, it's not a problem. We're going to do some of these questions that we're going over are involving cells or other things that I would think you would learn at the beginning of the year. I tried not to go to the later in the year stuff for the most part. Okay, so since you guys aren't saying anything, I'll go ahead and start telling you my thought process. So my claim would be something about how it would change the shape of the caspase 1, right? Because let's say it's shaped like a circle, but the part that it needs is something that's like jagged. When you cleave it, then you could get that jagged edge. So pretty much in terms of proteins, which is what caspase 1 is, structure is directly related to function. Like it cannot do what it needs to do without its structure. So that's what I would say. I would say something along the lines of cleaving inactive caspase 1 results in the activation of caspase 1 because it changes the shape of the protein. And then it says, provide one reason to support the student's claim of shortening the response time. So this part, really don't overthink it. Some of these, just as a general piece of advice for the FRQs on this exam, do not overthink them. Like you might read that and be like, what are they talking about? And start thinking all of this complicated stuff. This is literally the answer. All right, ready? Pretty much, it is faster to have one ready than to make one when you need it. That's it. Like it's not super complicated. When I read this and some of my questions last year, I was like, oh my goodness, what is this? And then I started overthinking and trying to come up with these brilliant magnified answers. But really, it's just something as simple as it's a lot faster to have the caspase 1 ready than to have to produce it when a bacteria comes into the cell. So that's just a general tip. We're going to go through a few of these questions that are going to look really complicated, but when you just break it down, it's very simple. Okay, so next, part C. A student claims, if you have any questions, make sure to put them in the chat. I will answer them to the best of my ability. A student claims that the NFKB protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. Identify 2 fractions where N-terminal Gasdermin would be found in cells infected with pathogenic bacteria. Okay, so when I was reading this question, I actually got a little bit confused. So I personally would do something like split this question into 2 parts, like in my brain or draw a line on my paper or whatever. So right here at the identify, because here, this first part before the identify is talking about the NFKB protein. But then the second part is talking about the N-terminal Gasdermin. So in my brain, I would circle those 2 words, the different types of protein and the Gasdermin, or draw some sort of line or put a little chart on my paper so I know that they're talking about 2 different things. because if you look at the Gasdermin for the first part of the question, it won't make sense. And if you look at the protein for the second part of the question, it won't make sense. So you have to kind of view them separately. So the first part, the student's claim, and you have to justify the claim with evidence that it's located in the cytoplasm. So that goes back to this faction part. So because up here, it doesn't give you any information about the location. The fractions are the part that give you information about the location. So it's asking about the NFKB, which is this last column. And it's saying is located in the cytoplasm. So what I would look for is something that I know is located in the cytoplasm. Or if I don't know what's located in the cytoplasm, I use process elimination for literally everything. And so I would use process of elimination to determine what's located in the cytoplasm. So if you guys have any ideas of what you think or what you don't think is located in the cytoplasm, put them in the chat and we can go over them. But I will start with my process of elimination. So ignoring the last column, because that's what we're trying to find, right? So we would say, so the way I would view this is if it's in with something else, and that something else is or isn't in the cytoplasm, then I would make my determination based off of what it is with, if that makes sense. So as we go through, it'll make a little bit more sense. So starting with the aconitase. Luckily, we don't have a verbal part of this test, so we don't have to learn how to say all of these names, but it's a Krebs cycle protein. So you guys might not have learned about the Krebs cycle yet because it's so early in the year, but it's a part of cellular respiration, and it occurs in the mitochondria. So it's only in the mitochondria. So that means it's not in the cytoplasm. So the immune response protein, the NFKB, is not with that, which means it's not in the mitochondria. DNA polymerase. DNA polymerase is found in the nucleus, and it's involved in transcription. But since we haven't gotten there yet, I'm not going to go too deep into that, but DNA polymerase is found in the nucleus. So, and also, no, it's found in the nucleus. So the NFKB is with the polymerase. So in my mind, I would be thinking, okay, maybe nucleus and cytoplasm. I wouldn't be sure. I would make sure to go through all of them. The GADPH is glycolytic protein. So glycolysis is another part of cellular respiration, and it actually happens in the cytoplasm. So I would say, okay, this is the one that I need to be looking at because this one happens in the cytoplasm. I would circle it on my paper. I would do something so I remember, okay, this is the one I need to be focusing on because this is the one that occurs in the cytoplasm. And then I would also view the sodium potassium pump just to be sure. Process elimination. I have to make sure all the other answers are not, involved. And sodium potassium pump is located on the cell membrane, not in the cytoplasm exactly. So here I would say this is the one I need to be focusing on because it's saying justify the student's claim. Where is it? Justify the student's claim that it's located in the cytoplasm. So pretty much I would just say something like, based on the chart or something like that, we're going to go over the scoring guidelines so you can see exactly what they want. But this is just something along the lines of what I would write. Based on the chart, the protein is located in the cytoplasm because it's located with the GADPH glycolytic protein, which is in the cytoplasm. Something along those lines. Also, if you have any questions about this chart, put it in the chat because there are a few charts like this that I saw last year when I was taking the class, like in my assignments and my multiple choice, just like in general. And so some of these can be hard to read until you get used to them. So if you have any questions about how to read it or anything like that or how to interpret it, please put it in the chat because it actually comes up a lot. I don't know why, it just does. So like not just this free response question. So that's the first part of the question. The second part of the question tells you to identify two fractions where the N-terminal Gasdermin would be found in cells infected with pathogenic bacteria. So that whole last part, cells infected with pathogenic bacteria, it just kind of fluff words because that's what this entire FRQ is about. So don't get too confused and be like, oh, well, is this cell infected? Is this cell not infected? Pretty much this FRQ is about cells that are infected. So the two fractions where the Gasdermin would be found, so this goes back up to the picture, right? So you have to find where's the Gasdermin. Gasdermin is here in step 3. So the Gasdermin is the one that forms the pore, which I believe lyses the cell. So then looking at the fractions, right? So going through what I think each fraction is, right? Based on when I did my process of elimination. Fraction 1 is the one that has the Krebs cycle protein, which makes me think it's in the mitochondria. So I would write next to it something like a little note to myself saying mitochondria or maybe a little drawing of mitochondria depending how much time I have, but something lets me know this fraction is mitochondria. Fraction 2 has DNA polymerase, which is found in the nucleus. So again, I would put a little note to myself saying this one is the nucleus. I would have done this on the last question, but since it's needed for this question, I'll just go over it now. Fraction 3, GADPH, which we established was in the cytoplasm, so I'd have written a little note beside fraction 3 over here somewhere that says cytoplasm. And fraction 4 is the sodium-potassium pump, which is on the cell membrane. So I would write a little note to myself, cell membrane. Okay, so it's asking where the Gasdermin would be found in cells infected with the bacteria. So if you look back at the picture, Gasdermin is step 3, which is the one that forms the pore. And if you look, this is cell membrane. So my mind would say immediately, okay, the pore forms on the cell membrane, the Gasdermin forms the pore, thus the Gasdermin would be found really, really close to the, if not on the cell membrane, right? That's kind of a logical step to make. So the one that I marked as cell membrane was fraction 4. And then it said identify 2. So again, what I would do if I was kind of unsure is process of elimination again. Because I would say Krebs cycle, this is that fraction 1 is mitochondria. So not in the mitochondria because that isn't related to its purpose, which is to make this pore on the cell membrane. Fraction 2 is nucleus. Again, if it's making a pore on the cell membrane, it has nothing to do with the nucleus. It has no purpose being in the nucleus. Fraction 3 is a cytoplasm. I would say yes, cytoplasm would be the other fraction just because it's what is inside of the cell membrane. So in my brain, I forgot what it's called, the Gasdermin would be floating around in the cytoplasm, ready to be activated, and then it's activated, and it does its thing with the cell membrane. So that's how my brain would work. And again, process of elimination can be really helpful with these questions. And so fraction 3 and 4, because they're the closest to the cell membrane, and also they're the only ones really relevant in this case, because it's not talking about mitochondria, it's not talking about the nucleus. So you have 50-50 chance if you've identified them correctly. Okay, part D. Describe the most likely effect of Gasdermin pore formation on water balance in the cell in a hypotonic environment. So have you guys learned hypo, and isotonic? If you have, then in the chat, see if you can try to come up with some ideas for how you would answer this question. So first, before going into the whole Gasdermin pore formation, I would say hypotonic. When the cell is placed in a hypotonic solution, what happens? Because you know all this tonicity has to do with water movement into or out of the cell, or no movement at all. It's just related to water movement. So I would say, okay, before I get into the pore formation, what happens when it's placed in a hypotonic solution? So hypotonic means that there is less solutes outside the cell and more inside the cell. In hypotonic environments, water rushes into the cell. So I would say, okay, water is going to go into the cell. So the effect on Gasdermin pore formation would be that water rushes into the cell would probably lyse, which means it just explode pretty much because there's so much water going into the cell. And pretty much I would say the pores wouldn't be able to form because it forms the pore and water rushes into the cell. The pore makes it even easier for water to rush into the cell, and the cell lyses and nothing can happen anymore because the cell pretty much dies. So that would be my answer. We'll go over the scoring guidelines, which is a little bit more specific, but in general, that's the general basis that I would go with. So for part E, explain how Gasdermin pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. So this part has kind of like two questions in it, right? Gasdermin pore formation and the interleukin release. So again, in my brain, I would kind of split it or circle the one separately so I know that I need to have two separate explanations, right? So Gasdermin pore formation, if you guys have any ideas, put them in the chat, but since you guys aren't being particularly active, although I think you should be more, we'll go ahead and start going through my thought process. So the Gasdermin formation, so it's asking about its effect on like when a cell is contribute to an organism's defense. So I would say when the cell opens, it allows water to come in, the cell lyses. And that destroys the cell, destroys the bacteria in the cell so it can't spread anymore, all of that. So that contributes to an organism's defense by just destroying infected cells so they can't spread any more bacteria to any other cells, pretty much. And then the second part, so that's my first little portion, 1 or 2 sentences. And the second part, how interleukin release contributes to the organism's defense. So interleukin, we haven't talked about in a little bit. It was brought up earlier in the question. Interleukin is the really important part here. This is why it's really important to read the paragraph that they give you. The interleukin, it tells you an interleukin is a signaling molecule that can activate the immune response. So that's pretty much what you need to say. So you would say the interleukin stimulates the immune response to attack the bacteria or to eradicate the bacteria or something like that. So that portion is pretty much given to you in the paragraph here. So that's, again, make sure you definitely read the paragraph, even though it can seem long and like it doesn't really, not really that important. Because otherwise, if you didn't read the paragraph, you would probably be very confused because there's no other indicator that that's what interleukin is. So we'll go through the scoring guidelines next. Again, the long response questions are worth between, okay, let me share you to my next tab. The long response questions are, there we go. The long response questions are worth 8 to 10 points. So you'll actually see the point breakdown here in the scoring guidelines. So I think it's this one. Yes. Okay. This was question. Oh no, it's not this one. We're going through a lot of questions here. Okay, so it's 2018. I don't know why it's not. Oh, here it is. Okay, so here are the scoring guidelines. Question 2. Describe the effect of the question that we did first, inhibiting step 3 on the formation of pores. So also what I would do when I go through the question itself and when I'm reading it is if I have time, if I'm in a rush, this is not like a 100% necessary step. So I would kind of try to get an idea of how many points each part is worth, because then I know, okay, I need to at least say something for this one because it's worth 2 points versus this part that's only worth 1 point. So you can kind of prioritize, because if you have absolutely no idea and this part is worth 3 points, at least just put something there. You never know. Versus if it's worth 1, you can prioritize and see what exactly you need to answer if you're completely lost, which hopefully you aren't, but if that happens. So part A is worth 2 points, and looking at the scoring guidelines really helps with this because you can see like the point distributions will kind of get an idea of how they're usually distributed. So describe the effect. So as Shreya said in the chat a little while ago, pores will not form. That's literally all you need to say. And then for the second part, interleukin release will not be affected. So again, don't overthink these questions. A lot of times that is how simple the answer is. Like I am someone who tends to overthink. So I will be looking at this and I'll be like, okay, what does it mean when the pores don't form? What does it mean when, no, that's not what it's asking you for. It's asking you for what the effect is on the formation. The formation does not happen. That's it. So part B was the part where we had to make a claim. Make a claim about how cleaving inactive caspase 1 results in activation. A student claims that pre-infection production of inactive precursors shortens the response time of the cell to infection. Provide one reason to support the claim. Okay, so the claim, the one that we decided on, was that it changes the shape or the protein, and it changes the shape because, again, shape is directly related to protein structure. They need their shape. Structure is directly related to protein function. They need their shape, they need their structure in order to do what they need to do. So again, it's very simple. Literally, cleaving means breaking, and breaking changes the shape. So, and then for the next part, reasoning to support the student's claim. So the student claims that the production shortens the response time. Again, this is the part that I said don't overthink. Literally, cleaving a precursor is faster than making one brand new when it's infected. So again, these questions, it's really important not to overthink, because sometimes the answer really is as simple as it seems. So part C, this is one with the student's claim, and this is one where we had to use the chart. So identify 2 fractions where N-terminal Gasdermin would be found in cells infected with pathogenic bacteria. So justify the student's claim about it being located in the cytoplasm. So the justification here was that it's found with the glycolytic enzymes. So I would go a little bit further in my explanation just to kind of be safe, because as I said at the beginning and when I was giving you some tips, just kind of write a little bit more just in case, because you never know, this justification, they could have wanted you to go a little bit further. So I would have said the NFKB and glycolytic enzymes are found together, and glycolysis happens and the enzymes are located in the cytoplasm, or something along those lines. So that's the answer there. And then identify 2 fractions where the Gasdermin would be found in cells infected with bacteria. So we said fraction 3 and fraction 4. It has a question up here. So we decide on fraction 3 and fraction 4 because it is related to the pore formation on the membrane, and pretty much needs to be very close to the membrane. So that's that. And then the next part, describe the most likely effect of Gasdermin pore formation on water balance in the cell and hypotonic. So water enters the cell. So I went a little bit further and I had said something about the cell lysing. Like you don't even need to go that far. You literally just need to say water enters the cell. This is how simple the scoring guidelines are, and this is how your AP readers score your free response question. So even though this question is a little bit weird because it does say the effect of the pore formation, but since there's pores, water enters the cell. You don't even need necessarily to mention the pores because it's given. And then for part E, explain how the pore formation and interleukin release contribute to the organism's defense against a pathogen. So as we said, the pore formation allows the cell to lyse, which again means it just kind of explodes, which destroys the cell, obviously, because it explodes. And the interleukin signaling stimulates the immune cells to destroy the bacteria, because as it gave you in the little description, that is what interleukin does. So the description is really important. So in terms of point breakdown here, you have this one, it's 2 points because it says this and this. So I would have guessed around 2 points. This one I would have also said is 2 points because it says make a claim and provide 1 reason. This one I would have maybe guessed, oh, it is 3 points, never mind. This one is 3 points, justify and identify 2. It's really not that hard to kind of guess the point breakdown. This one is only 1 and this one is 2. So in terms of prioritizing, if you need to, hopefully none of you need to do this on the exam, but who knows. So in terms of prioritizing, I would have said, okay, I need to answer A, B, C, D. If I don't know, I can skip that one, or if I have something else I need to get to, or if I'm running out of time, I know that that's not the most important, that's not the most heavily weighted. But ideally, you don't want to have to do that. Okay, so we're going to go over some short free response questions as well. So let me pull up the next one. The next one we're going to do is, for the short one, is 2017 number 5. The short ones tend to be very short. So yeah, it's only 4 points, I think. 4 points. So this one says Microcystis aeruginosus, again, thank God we don't have to pronounce these, is a freshwater photosynthetic cyanobacterium. When temperatures increase and nutrients are readily available in its pond habitat, undergoes rapid cell division and forms an extremely large visible mass of cells called an algal bloom. It has a short lifespan and it is decomposed by aerobic bacteria and fungi. Identify the metabolic pathway and the organism that is primarily responsible for the change in oxygen level in the pond between times 1 and 2 and between times 3 and 4. So here, if I'm looking at the graph that they give me, I see that it's relative concentration over time, and it's concentration of oxygen. So here I'm looking at, I see that oxygen goes straight, goes up a little, peaks, and then starts to come back down after around time 2. I see that the cyanobacteria goes up, peaks around time 2 as well, goes down, decomposers go, and they peak around time 3 and then go down. So just a general, how I would interpret this graph generally, because again, you do have a time restraint. So it says identify the metabolic pathway and the organism. So what I would do is first look at the organism, and then from there, if I'm not sure, I can just say what pathway does this organism use. In terms of metabolic pathway, it means photosynthesis or cell respiration. So don't get scared by those big words. So between 1 and 2. So now I'm only looking at 1 and 2, so I'm ignoring the rest of the graph. Between 1 and 2, I see that the oxygen goes pretty flat, and then it increases and peaks. That's all it does from 1 to 2. And then now I look at the cyanobacteria. Cyanobacteria also goes flat, and then increases and peaks. Decomposers stay pretty flat the entire time. So in my mind, I say decomposers isn't changing, so they can't be responsible for the change, if that makes sense, right? So since the decomposers, nothing is going on with them, so how could they possibly be responsible for a change in anything? So that leads me to believe the cyanobacteria are responsible for the change. So from there, I say, okay, so the organism is cyanobacteria. What's the pathway? So bacteria can do photosynthesis or respiration depending on the bacteria, but luckily they gave it to you here. It is a photosynthetic cyanobacterium. So it's telling you that it conducts photosynthesis. So that's already 2 of the 4 points for this question, right? You have cyanobacteria and you have photosynthesis. So between 1 and 2. So you have to specify, obviously, that that's between 1 and 2. Photosynthesis is the metabolic pathway, and cyanobacteria is the organism primarily responsible for the change in oxygen level. First sentence. Second sentence, between times 3 and 4. So now I'm only looking at 3 to 4. And I would say, okay, looking at oxygen is constantly decreasing here. And then looking at cyanobacteria now is flat. And decomposers is decreasing. So based on the same logic that I used for the last question, I would say decomposers is decreasing and oxygen is decreasing. Nothing is happening with cyanobacteria, so how could they be involved? So then I would say decomposers are responsible for the change, and then decomposers conduct cellular respiration. They don't conduct photosynthesis. So it's really the short answer questions. That's all you have to say. That's 2 sentences. And also for this question specifically, sometimes you can just kind of use guessing, right? So if you get this first part right, I would say you'd have a 50-50 chance, right? Because you could say decomposers are responsible and they do respiration, or you could say cyanobacteria are responsible and they do photosynthesis. Hopefully you don't match them up wrong because it gives you here that it's photosynthetic. So it really is that simple. And honestly, if I had no idea, I would guess, and I'd have a 50-50 shot because I would have matched them correctly because it gives you that it's photosynthetic. And then it's just a matter of which one you match to which time period. And if you get the first one, you should get the second one because logically they're not going to ask you the same one twice. Like, that's just a guessing tactic, right? They're not going to ask you and expect the same answer to the same exact question twice. Like, it's not going to be cyanobacteria and cyanobacteria. So in terms of that question, so these questions you would have 10 minutes on. But since you have less questions than they did in the past year, so this is the 2017 exam, so they had 8 total free response questions. So if I had to guess, I would say your exam these might be a little bit longer, but not much because they're still short questions. They are called the short response questions. So I would say it might be a little bit more complicated or a little bit longer than this, but not like that first one that we did. That was a long question for a reason. So we're going to go over the scoring guidelines for this one. For this one, the scoring guidelines are very short, just like the question was very short. So it's not particularly like in-depth for the scoring guideline. So here it is. This was question 5. So it says scoring guidelines. Sometimes I format it like this. It just so the readers can pretty much just check off boxes and say, okay, they did this, I'll give them the point for this. Very simple. Makes it easy for them and easy for you to get points. So time period 1 to 2. The pathway is photosynthesis. The organism is cyanobacteria. Or you could have written the long name, but you didn't have to. Pathway 3 to 4. The pathway is cellular respiration, and the organism is decomposers based on the graph. Or if you wanted to go more in depth, you could have said fungi or bacteria or something like that. So this scoring guideline wasn't particularly in depth or deep because it was a pretty straightforward question. There's no like intricacies to look at. So we're going to look at another short response question. And then I think we will have time to look at a part of a graphing question. So the next short response we're looking at is 2019. This is the question that I took when I took the exam last year. So here it gives you. The petal color of the Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of petal cells. Petal color is determined by the pH of the vacuole. A model of a morning glory petal cell before and after flower opening is shown in Table 1. So that's what it's giving you. It's telling you before and after the flower's opening. And when you look, the difference that happens when it opens is it changes from red to, the petal cells swell and it changes from red to blue. So I would see that the pH of the vacuole increases, the flower color changes, and it just like gets bigger. And if I were to look at analyze the actual picture, what I would pick up would be clearly it's bigger. There's more potassium inside in the open flower, and there's more hydrogen outside. So the hydrogen left and the potassium came in. But that's pretty much all you would need to get from there. So identify the cellular component in the model that is responsible for the increase in the pH of the vacuole and describe the component's role in changing the pH of the vacuole. So how I would approach this, since you guys don't want to be active in the chat, but if you guys have any ideas, feel free to put them in the chat and we can discuss them. Or if you have something that you think was right, but it turns out to be wrong, put it in the chat and we can figure out why it ended up being wrong. So how I would approach this, right, is saying, what changed the pH? I took AP Chem, so I know that pH is potential for hydrogen and it's related to the hydrogen ions. But even if you didn't know that, you would look at the two different vacuoles and see what's different. This one has more hydrogen ions, this one has less, and this one has more potassium. What is responsible for that change? The transport protein, the potassium hydrogen transport protein would be responsible. Proton pump, you see the in arrow. But clearly the hydrogen ions left. So if the proton pump is pumping it in, that can't be responsible because how'd they get out? Whereas when you look at the potassium hydrogen transport protein, you see that the arrow is pointing out. So that's what's taking these two hydrogen ions and pumping them out, is pumping the potassium in, which is how all of this got in here. So pretty much you look to see what changed and then see what is responsible for that change, if you didn't know any details about what pH is exactly. So identify the cellular component. The cellular component is the K+/H+ transport protein, and its role is that it pumps hydrogen ions out of the vacuole, and that's it. I wouldn't go into talking about the potassium ions because pH is directly related to hydrogen ion concentration, not necessarily potassium. So you want to stick to talking about the hydrogen. I'm sure if you talk about the potassium being pumped in and the hydrogen being pumped out, you wouldn't get points off, but it's best to just stay talking about the hydrogen just in case so you don't make any silly mistakes or contradictions. So part B, a researcher claims that the activation of the KH transport protein causes the vacuole to swell with water. Provide reasoning to support their claim. So yes, that's what we established in part A. So I would say, okay, the researcher's right. My reasoning would be clearly based on tonicity, right? When you learn that there are more or less ions in solution, more or less solutes, that means that water is going to be pumped in or out of the cell. So I would say that the transport protein causes the vacuole to become hypertonic to its surroundings. So hypertonic means that there are more solutes. So in this case, I would say the solutes would be the potassium. But even if you want to look at potassium and hydrogen, in general, there's a lot more solutes, 1, 2, 3, 4, 5, 6, 7, in here than there are in here, which is only 3. So whether you want to look at the potassium, which is the most obvious one to look at, or even if you want to look at them combined, there are a lot more in the open flower, this one that's swelling, than the closed one. So from there, you'd say when the transport protein is activated, water flows in because the vacuole becomes hypertonic to its environment. Or even if you don't want to say that specific, you would just say as more ions are introduced, or more solutes, which are ions, are in this case, are introduced into the vacuole through the pump, water flows in, and the vacuole swells. So even if you don't get as specific with your tonicity, if you're a little bit confused, because that can be a confusing topic with all the different prefixes and prefixes, either way, you don't necessarily have to use the word, but using the word sometimes can be a keyword for the readers that can be like, okay, she said hypertonic, check, he gets the point, right? So in that case, I would try to use the word. So, so again, these are short questions for a reason. This was only an A and a B, and the B was based on the A. So if we want to try to guess the point breakdown for this, so if you guys want to try to guess it in the chat before we go to the scoring guidelines. So for part A, in terms of identify the cellular component and describe the component's role, what do you guys think the point value would be? These are worth 4 points total. So actually, this one, I'm not sure about. This one is worth, I'll tell you what it's worth, and then you can get it from there. This one I think is only worth 3. This one is only worth 3 points. So hopefully you guys can get it from there. But please put it in the chat what you guys think. And then we can go to the scoring guidelines next. So for parts A and B, the total is 3 points. So this one should be pretty easy because I told you the total. But I'm going to switch to the scoring guideline. So scoring guidelines, this was 2019. Okay, so here it tells you it's 3 points. Identification was 1 point, description was 1 point, and reasoning was 1 point in part B. So for part A, as we said, the transport protein is responsible. That's the identification. You don't have to go any more in depth than that. It's really saying what is it? It is the transport protein. Description, it moves hydrogen out of the vacuole, which is what we said as well. And then for part B, you have your reasoning. You could say the concentration of solute is increasing. In the solute, you could say the concentration is increasing. Or you could say the solute is moving into the vacuole, making it hypertonic or hyperosmotic or lowering the water potential. So either of those answers would be fine. And that question is a short question. So it's on this one it's only worth 3 points, but for your exam this year, College Board said they would all be worth 4 points. So since we have a few more minutes, again, if you have any questions, please put them in the chat. I'm going to go to just one specific part. It's part B to look at how to graph, because there's going to be at least one graphing question in every exam. So they can be kind of scary, but in reality, they're also very simple. Again, it's important not to overthink. That's my major tip for you. So we're back at 2018. This one is 2018 number 5 here. Okay, where is it? No, that's the question. Okay, so some birds, including great spotted cuckoos, lay their eggs in the nests of other birds, such as the reed warblers. The warbler parents raise the unrelated chicks and provide them with food that would otherwise be given to their biological offspring. A researcher conducted an investigation to determine the type of relationship between warblers and cuckoos in an environment without predators. The researcher found that nests containing only warblers were more likely to be successful than nests containing warblers and cuckoos, data not shown. A successful nest is defined as a nest where at least one chick becomes an adult warbler. In some geographic areas, several species of nest predators are present. Researchers have found that cuckoo chicks, while in the nest, produce a smelly substance that deters most predators. The substance does not remain in the nest if cuckoo chicks are removed. Figure 1 shows the probability that nests containing only warblers or containing both warblers and cuckoos will be successful in an environment with predators. In a follow-up experiment, the researchers added cuckoos to a nest that contained only warblers, group 1, and removed cuckoos from a nest containing warblers and cuckoos. So some of that information you need for the other parts, we're not going to go through those. We are going to focus here on part B. On the template provided, draw bars in the appropriate locations to predict the relative probability of success for the nest in the presence of predators where the cuckoos were added to the nest containing only warblers and the cuckoos were removed from the nest containing both warblers and cuckoos. So first, let's look at this graph up here, right? So first, you have to analyze what they give you. Well, after or before, it doesn't. I like to analyze after because then I have some context, but some teachers might tell you to do before and after, whatever works for you, honestly. So probability of success, with warblers only, they have about a 4.4, which is 40% chance of success. Warblers and cuckoos, they have about a 60% chance of success. So here, this is where they're asking you to draw the graph. So it might look a little bit funky or scary, but in reality, this is a really simple question, right? Warblers only and cuckoos added. So my brain would say, okay, that means warblers and cuckoos, which means the probability would increase, right? Because this one already has a high probability. You know that when they're together, probability increases. So if you have one and you add the other, your probability is going to increase. So you would literally just draw a bar that's higher. And you don't need to do any calculations to provide a specific number or anything like that. Literally just a bar that's higher than this one. If you want, you can make it match up with this one. It doesn't really matter. And then for this one, it would be the opposite reasoning, right? This has warblers and cuckoos. When you take out the cuckoos, what you're left with is warblers only, which is significantly lower than the warblers and cuckoos. So you would just have to draw a bar here that's lower than both of them together. So we can look at the scoring guidelines for this part, but the graphs seem like really scary just because sometimes you don't really know what exactly to do. But when you think about it logically, the graphs are usually very simple. So I'll show you the scoring guidelines for this one, and then this is the last one we'll do. But I'll show you the scoring guidelines so you can see what they're looking for, but it's very straightforward. And the only thing that I think would get you on a question like this is overthinking. So this was question 5, so let me go down. Okay, so here, graph. Cuckoo added, bar must be higher than the warblers only. Cuckoo removed, bar must be lower than the warblers and cuckoos bar. So it doesn't even, you don't have to do any calculations. It doesn't matter where it is when it's higher or where it is when it's lower, just higher and lower. That's all you need. So while it may seem really like intimidating, the graphing questions, that's the best word I can think of, they are intimidating. They tend to be very simple because there's such a wide range of people in AP biology. Some of them are good at math, some of them have had more exposure to math than others. So in terms of graphing, they can't do too much in depth with the graphing because of the different levels of expertise that the students have in AP biology. So, oh, I got a question. Can you assume that the protein is found, is found with the GADPH protein? So, in that question, I'm sorry, I was going to look at that at the end, but here, that was with this one. So the reason why you can assume that it's found with the GADPH protein is because they say that it's in the, because here actually, you have the plus and the plus. That's how you would read these charts. So you have, they're all found in the entire cell just because they're found somewhere. Fraction 1, this one is positive. Fraction 2, this one is positive. Fraction 3, the GADPH and the NFKB are positive. And fraction 4, this is positive. Sorry I didn't get to that earlier, but did that answer your question? I'm hoping. So if you guys have any more questions, we have a few more minutes, but otherwise, that will be it. So if you have any more questions, please go ahead and ask them in the chat or in the Ask a Question box, and I can answer them for you. It can be general about AP biology, or about the exam, or about FRQs, thought process, if you have anything specific about any of the ones that we did here, or anything like that. Okay, so if you were feeling not so great about the FRQs at the beginning of the stream, if you could put in the chat on a scale from, we'll go with 1 to 10, how you're feeling about them now. Okay, I also just got another question. Do you have any tips or resources for self-studying if your teacher doesn't teach? So I would say there are some videos on YouTube. Bozeman, I don't have to share my screen anymore. I don't know why I'm still staring at. He has a lot of really good videos that kind of go in depth and explain. And also I bought one of those review guides, and there's also a lot of them online that you can find like PDF copies of. You don't necessarily have to buy one if you can't. And for me, the most helpful thing was taking practice tests. I found as many practice tests as I could possibly even think of, and I would go through them and be like, okay, I don't know this. So then I Google and just find out what it is that I don't know. For me, practice tests were a really good way to kind of gauge where I'm at. So if there was something that my teacher didn't teach or that she missed, I would say, okay, this is on the test. I have no idea what this is. Let me do more research and find out. And I also took some really detailed notes, and the notes also helped me because then when it, both from the practicing and also in these streams or anything else that you're watching to use, taking notes is really important, because then at the end of the year in May, you can go through those notes and be like, okay, I remember I had some issues with this, or here's a helpful tip I had written down. It just helps. But I would also say definitely practice tests, practice questions, practice anything you can find, because that will be really helpful in kind of identifying what you know and what you don't know. So I'm kind of hoping that that answered your question. In terms of resources, and I would just say, honestly, just Google it. There's so much on the internet. But also, 5 Bullet's a great resource. We have weekly streams all the time. So yeah, hopefully that answered your question. Okay, so I didn't get any more questions in the chat or in the ask a question box. So I think that's it. Thank you guys for joining. I hope that I helped and kind of clarified the FRQ for you guys. Please tune in to our next stream, our next bio stream. I'm not sure when it is, but you can see. And yeah, thank you. Bye, everyone.